measure the concentration of platelets that is still present in the plasma, because it tells you something about the efficacy of the centrifugation procedure as such. It tells you something about, did the protocol perform correctly? If there's high concentration of platelets left, you know it's a confoundant because it's difficult to separate from the vesicles, etc. Now, it took another 4 years before we finally got this in a paper. It's a long time. Don't know why it took so long, actually. We've had many discussions about it. But what we published, I think it was last year, is a paper called My Blood EV. And we put there a checklist in for people, what we think may be very helpful for the future to better compare the quality of the prepared plasma, the serum sample. And these are the slides Fabrice prepared. He just sent them, so I will go quickly through them. So what everybody is doing now, when you write the method section of your paper, you tell that you collect blood using this needle, you use citrate as an anticoagulant or ETA or whatever, and then you have a centrifugation protocol, etc. We would like to keep that in place. That's why we have the checklist, but we would like that you also try to measure, for example, the concentration of platelets, report the hemolysis and the fat. That's all. And we think that by reporting these 3 additional parameters, at least for blood, of plasma and serum, you can really improve the quality and the comparability of downstream research. So we make a little list. This list has changed already a couple of times. We try to make it as easy as possible. And what we would like to do is that you do not only fill in this list, but that all these lists are being somewhere stored in the virtual biorepository, so that with Insight we can look at how do you say that, about the data, and that you can use these data to improve evidence-based the protocols that we use for plasma preparation and serum preparation and blood collection. So the idea is very simple, just implement a few quality controls if possible. Sometimes it's not possible, that's also fine, but then you can also report it's not possible. And there's quite some interest for this. So we are now in the phase of preparing several papers for societies. I'm one writing for the ICH, for example. We think that perhaps the idea of this reporting tool can also be used for other body fluids, so maybe it's not unique for blood, but perhaps you can also think of quality controls for urine or CSF. And now, right now, we have had a discussion with people from the NIH for a while. People from the ICH will be presented this data. It's very simple, but people have an idea that this may help to improve the quality of biobanks and downstream analysis. This is a hobby project from Fabrice, but I think it's extremely important. He would like to make a database where we store all these data and that we use in the future to optimize our protocols and to see whether it really helps to improve the data comparability. This is a study that I'm completely not involved in, but it's from, I think Fabrice is the leader together with Fazer Daniel, and they have an international biobank study ongoing where they try to identify most used protocols, and they are doing a head-to-head comparison of the plasma and serum quality. Of course, again, the only goal of all this, I think, in a sense, very simple idea is that we would like to improve the quality of the samples that we use as a starting material for blood EV research. Thank you. So, I think let's use our time now for discussion, because I think that's the most valuable part of this meeting. It's a sort of informal meeting, and I would like to hear from you what your ideas are, whether you think it's a good idea. If you have ideas, the blood EV task force should work on different topics, this is your moment of, well, opportunity to share your thoughts and ideas. Who will open the discussion?
Yeah.
Yes, sure.
Thank you so much.
I cannot hear you.
Oh, okay.
Now I can hear you.
Thank you so much. I have just several questions, but I want to focus some. It's very I used with the clinicians, and I saw several times urine sample reflects the blood conditions. Sometimes some sepsis patients shows so much amount of the gram-positive contents in the urine, or sometimes like that. So I think the relation between the blood and the urine based on the really standardized some collections, and then some compare each other, maybe giving us a new insight regarding some disease or some patient conditions. Is there any some collaboration between the blood and the urine groups to see?
Well, I think Uta is here and she can elaborate on this. I think what we see now is for several fluids, also for conditioned culture medium, people have been trying to make lists. One of the things, coming back to the first part of your question, we published a study, I think 1 or 2 years ago, a very simple thing that was more a coincidence than that we really planned a study, but we simply took the plasma samples from 3 institutes that use the same protocol to prepare human blood plasma, and we just measured the concentration of platelets. So people use the same protocol with 2 spins in 3 different institutes. Very well-known institutes, all 3. And what we saw is that the platelet count is very different between the 3 institutes. Actually, we even saw that the platelet count in plasma samples between operators in our own lab differs. And what we did, for example, in our own lab, we introduced a Lego brick, and we used the Lego brick to mark precisely 1 centimeter above the Buffy code on the platelet pellet. So people are not allowed to pipette below this 1 centimeter. That really helped in our lab. But the sense is that using the same protocol does not guarantee you that the end product has the same quality, consistency. And I think that was quite an important lesson we learned. And why is this so important? You can think, who cares about these few platelets? The concentration of platelets we have in Amsterdam is about 100-fold below the platelet concentration that can be measured by a routine hematology analyzer. So if you measure the concentration of platelets in our plasma sample, the analyzer will tell you it's 0. That's not true. We still have about 1 million platelets per mil. The total volume of this 1 million platelets per mil of plasma, if you compare it to the total volume of the vesicles, is about equal. So now our plasma is relatively very pure to most other hospitals prepare regarding the quality of the plasma. But if we remove these few platelets by a filter, the concentration of EV-associated microRNA dropped 50 to 75%. So even if you have a very ultra-low platelet concentration, it already has a dramatic effect on your EV-associated microRNA. So when I then look at the literature, and we looked at the 20 most recent papers studying microRNA associated with vesicle in clinical studies, I think 1 in 5 did not report anything about blood collection handling, and the other ones were all completely different. So, and then you know something is really wrong in the basics.
So regarding the platelet contaminations, I also have additional questions. So there is so many some biobank exist in each countries, and I'm really happy to see that this international challenging project. However, I also want to know that is there any some project to deal with already stored plasma to minimize the effect of the platelet to derive the EB or something?
No, not to the best of my knowledge. No, but it's a good point. But I think this is all not rocket science. This is all very simple. But I think by simply improving, or at least knowing the quality of the end product of your biobank with the starting material for EV research, you can greatly improve the comparability of downstream results from your EV studies. If you now look at most of these studies, what people have done, at least for blood, all these data are really single-center studies and incomparable amongst each other, whether it's omics or you name it. We think that by at least trying to improve or knowing the quality of the starting material for your EV research, I think it's one of the major factors confounders of your downstream analysis. IOBankers use the term from bench to bin. I don't know whether you're familiar with that one, but I think that summarizes it in 4 words. Yeah.
Thanks. I have 2 comments and 1 question. One is, I know it's focused on human, but many of us do both human and animal work. So it would be very helpful to add animals to this. Time of day is critically important, fasting is critically important. And then number 2 is, while I appreciate all of these different guidelines, it would also be helpful to have some suggestions, for example, how to measure best for lipoprotein content, because again, you'll see a paper that has using ApoB, another paper uses something different. So that's actually my question. So first, it'd be nice to have some concrete guidelines, and then the question is, what does the study group think about as the best way to measure lipoprotein content?
Yeah, well, both are, of course, very valid. The first, regarding animals, I think we simply started with human blood because it's mostly used. But of course, you can do essentially the same thing for mouse blood or dog blood or whatever. Only thing, sorry?
I just think it should be added to the guidelines.
Yeah. That's a valid point. The other thing is, what's the best technique to measure? Well, I think like lipoproteins, there are many different ways to quantify lipoproteins. So, 2 things. First is, I think, we do not say that you cannot use samples from existing biobanks if you do not know these parameters, but at least then report that you don't know them. If you do a new clinical study with, let's say, 1,000 patients, I think it would be ridiculous for all 1,000 to determine these parameters. But let's say pick 10 randomly or 100 randomly, whatever, and look what the quality and the consistency is of the samples that are being prepared. Just to give you an idea, we shared our blood collection and handling protocol to prepare plasma with many different centers abroad. We always ask them to collect blood and prepare blood plasma using our protocol, and then send the frozen plasma samples to our lab to check for the presence of platelets. Just that we know that once people are doing the real clinical study, that we are not looking at all sorts of artifacts. And that really helps, because sometimes it goes perfect, sometimes it's crap. But you better know it beforehand. So, as long as people are becoming aware that perhaps it is wise to put a bit more attention to the quality of your starting material for EV research, how you determine the lipoproteins, for example, I think that would be my second problem. I think I would be already very happy if people at least tried to look at the lipoprotein content. And I think actually that Fabrice was working on this, so I will take it with him to discuss it. Yeah.
Hi, I'm Srivastava from Thermogenesis Industry. So one of the questions I have is that we don't see, when you talk about blood EV, we don't see so many studies where we talk about donor variability, such as male versus female, old versus young, within female, reproductive age female, and versus postpartum female. So I was wondering if the task force has some plans to go, or is it worth including that in
Yes.
the overall estimation?
Yes,
Thank you.
well, that's a real good question as well. So I can just tell there that we're just finishing a very large manuscript where we show that it's possible to measure the same concentration of a particular type of cell-derived vesicle in blood plasma across different instruments, in this nowadays flow cytometers. That has been Now, I think 3 or 4 years of work in total, but the answer is yes, we can measure the same concentration of vesicles in a given sample, keeping in mind that the vesicles are very heterogeneous. Vesicles are the only element present in blood that are very heterogeneous in size and do not have a unique surface marker. So that really makes comparability even more complex. But it is possible to measure the same concentration of a particular type of vesicles, let's say platelet-derived vesicles. And now I forgot your question, thanks to the jet lag. What was the, oh yeah, now I know, the effect, sorry, the effect of age or whatever. So what we did is we thought one of the things that people, at least in our clinic, but I guess worldwide use most, is a reference range table. So if clinicians suspect somebody from inflammation, they will ask to measure the concentration of leukocytes in your blood. Why do they do that? Because there's a reference range known for leukocytes. Concentration of leukocytes is in a certain bandwidth. If the patient, if the blood sample contains a concentration of white blood cells way higher than the reference range, the clinician thinks, well, it's more likely that this patient is, let's say, developing sepsis or is suffering from inflammation. So if you go through the literature, and also the data we published ourselves on the concentration of blood EVs, it really is variable. Even in one lab, as in Amsterdam, we have published all sorts of concentration over the years, and they all share. So now we can calibrate flow cytometers, we can generate absolute concentration of vesicles. For the first time, we can compare the concentrations of certain vesicles between instruments. And what we now did, we collected blood from, I don't know, 200-300 healthy individuals, and we have determined a reference range of vesicles that we also added to the manuscript. And we are also looking now at what's the effect of age, what's the effect of gender. And actually, at least I can tell you that regarding gender, we completely see no effect at all. Zero. That will not be in the first paper, that will be hopefully in the paper that we publish later on. And age, I forgot, but I think also there was not much effect. So, but at least for gender there was not. But I think we need some data sets that really we know what has been measured. It should be in absolute units and it should be clinical useful. And I think once you have these data, in this case for, let's say, red blood cell vesicles or platelet vesicles for blood, then you can go to patient cohorts and see whether, for example, cancer patients have average concentrations of platelets. There have been tens of studies published, but they all have different concentrations that were reported. And the value of these single center studies is nil. Clinically, you cannot do anything with it. You need to have comparable data somehow. So,
that's good. Felicity Dunlop from Deakin University. So I have a question and I guess a comment. So we've started working with mouse plasma, and you can appreciate you get a lot less plasma out of a mouse as you do a person. So I think I'd like to see the guidelines include what's appropriate for a mouse. So you can't sort of leave the top centimetre above your buffing coat because that's your entire sample. And I guess the other one is, so we haven't started measuring platelets yet, or do we do a lot of centrifugation steps to remove them? Do you have a recommendation on the method to measure residual platelets in your plasma samples?
what was your first question?
So we're working with mouse plasma.
Yeah.
So you only get a small volume of plasma out of a mouse. So maybe 500 microliters if you're lucky. So in the guidelines that have been written, it'd be nice to have specific guidelines for small animals. So for instance, you can't leave a mil of plasma because that's all.
Yeah, so this refers back to the earlier question. So I think I don't care, nobody cares whether you have platelets still in your plasma. That's your problem. But I just would like that if you report your data, that you also report that there are still vesicles, still platelets present in the plasma, and if you can report the concentration, that would be very helpful. Because what happens now is that everybody measures nothing, so we have no clue. We only have the protocol, what people have been doing to prepare the plasma or serum or whatever. And again, the quality is very different, even with the same protocol for human blood between centers, and for mouse it will be exactly the same. So at least, as long as you're aware that you may have confounders and you try to quantify them and report them, that already would be a massive step forward.
Yes. So we're
Because, for
using-
example, if you would do size exclusion chromatography or density gradient or whatever, I have no clue, at least then you know that it's unlikely that you separated the platelets from the vesicles, and you should be careful with the interpretation of your data. And you should not say that it's a microRNA associated with the vesicles, but perhaps in an EV-enriched fraction.
So we're using proteomics as a readout, so we get a lot of data. So for instance, we can measure lipoproteins directly. We also get platelet markers. So some of these sort of smaller scale experiments where we get huge amounts of data, it might be worth trying to consider incorporating marker proteins and ratios, which is what we're starting to look at now. So we get a really good purity indication of like, yeah, there's still a heap of lipoprotein in there, but we can reduce intrasample ratios to EV markers.
Well, I can tell you that there's now, it's an idea from Michiel Pechtel, and he's writing a grant on it, and I'm sideways involved in that. What he's trying to develop is based, I think, on RNA sequencing, that when we have samples in the existing biobank, and we can do an RNA analysis there and look how many quantify via that indirect way the contamination with certain cell types and correct for it. And I think perhaps you could do something similar for your omics. So it's correction afterwards. But I think the point more is that it would, I think the main goal of my blood EV is simply that we increase the awareness that the starting material samples are often not perfect, which is fine, that's no problem, but at least be aware of that and try to quantify it and report it. Be transparent about that the samples are never perfect, no problem, but it would be good to know because later on perhaps you should be more careful with data interpretation, but also between interpretation of data between the centers. I mean, if you see the same, people have done the same work, but one has, let's say, 100-fold more lipoprotein or platelet contamination than the other, then perhaps you should be more careful with interpretation of those data than with studies where they have very clean samples. Now we have no insight. This is completely blank.
Hi, I'm Abha from the University of Michigan. My question is about using healthy controls and comparing that with, say, a case study like you have diseased. And what are the right parameters to standardize this, or how do you normalize it? Do you normalize it based on the concentration of EVs, or the volume of plasma, or the housekeeping genes? What are like the best techniques to do that?
What would you like to normalize? I don't get the question, sorry.
So if I'm comparing, say I'm doing a case study for a particular kind of disease, and I'm comparing my patient cohort with some healthy controls, and I see, as you mentioned, the variability in donor to donor type, and which some other people also mentioned. So how do I normalize this? Like what I'm looking, sometimes I tend to forget the question I'm asking because there is so much heterogeneity in the data I see. So how to go about it?
Well, that's a real good question. So, from my experience, what often happens in our hospital is that let's say, if in the cardiology samples are being collected, and the control samples are being collected somewhere else, then usually the quality of the prepared plasma or serum samples is very different. How to normalize it, I have no idea, but I think if you would again measure simply platelet concentration, etc., at least you have an indication that the sample quality is consistent or not. If there's a huge variation or a huge difference, you have a problem, but better know that and report it than just to whether it doesn't exist. There's a nice example of Professor Takahiro, who was, I think he's doing all sort of microRNA study or RNA studies on vesicles in blood. Thousands of samples, and actually he's looking at cancer and whatever. But the interesting thing is that after analysis of thousands of samples, AI, you name it, the biggest effect is the center where the sample was collected. That has more effect than everything else. So yeah, that again shows that how the sample is collected and processed, yeah, that determines the composition, and the composition affects your downstream analysis. So as long as you're aware that the sample quality is perhaps not consistent and may differ between the patient and the controls, just be aware of it, try to circumvent it, and otherwise at least report just try in a clever way to see whether these samples are comparable regarding quality or not. I think that's the most important lesson, because there will be differences. But that's not so important, but at least be aware and try to report it. If your patient samples have more platelets, fine, but report it. Because now we do the same, but we do not report it, and now we have incomparable data in the end. So you have to start somewhere.
Hi, this is Sheng Yuan from Nanyang Technological University in Singapore. So I'd like to share that I face quite some challenges when studying mouse blood. The reason is we're working with different animals specialists, and they have different ways of extracting blood from mouse, and especially when we need to study more than 400 microliter of blood, so usually we use cardiac puncture. But when we try to communicate across different labs, how to do cardiac puncture without video is extremely challenging. And most of the time, the blood clot immediately, and hemolysis rate is more than 50 to 70%. So we even, because of this problem, we developed microfluidics device to isolate plasma in a much gentler way and faster way, but we still observe hemolysis. So do you face similar problems or have some guidance for the extraction?
I think I don't know anything about mice because I hate animal studies in general. But what I do know, I think mice have a different hematocrit than humans. And so if you collect human blood with an anticoagulant, the anticoagulant will only dissolve in the non-cell fraction, so in the fluid fraction. So if mouse have a larger fluid fraction, you should use more anticoagulant.
Yeah, the amount of anticoagulant is quite tricky because usually we will pre-inject or rinse the syringe with anticoagulant. But the amount of blood we can extract from mouse varies a lot. Sometimes it's 200 microliters, sometimes it's 400. So the amount of pre-rinse ETTA is kind of like blood to anticoagulant ratio is not constant.
Well, again, I think, I don't know how to solve this problem for you, but I think as long as you're aware of the limitations and report that you have hemolysis and perhaps that there was a clot during the collection, at least report it, because that makes it more fair. But if you have such small quantities, I think it will not be easy to solve all your problems easily. I mean, 400 microliters isn't that much.
I would like to happy to report, but so far all the blood hemolyzed, so I cannot report.
Yeah, but if you go back to what you published in other people, is it always reported there is hemolysis in these samples? Do people report it?
Come over here, we'll talk about it.
Sorry, what?
Okay, thank you very much.
So what did you say?
We know how to do mouse blood.
Okay, well, your problem solved. Oh, that's even better. So.
We should all talk about the life of
So actually, I also want to join that school. And another question. So many some confounding factor maybe exist in the human, also the rodent or other animal studies. So is there any some platform to share the some potential confounding factor I'm interested in the some circadian rhythm, which may affect some result of the blood biomarker or something. Also some yes, it might be like kind of some crazy idea to see the seasonal changes because our like some entire body is regulated in the autonomic nervous system to adapt each like the season. I think like these like some conditions also may affect kind of some biomarker derived from like some blood or something like that. So I think it's very important to share the kind of unsolved, like some confirmed factor, what is already known and what is unknown, but need to be solved in the future. So,
Yeah. Well, actually, you can summarize all this simply in transparent reporting. I think nobody cares what you're doing, but please report it, including the quality of the software. I think that is the lesson that would make life easier for us after. Keep in mind that I think that in the physical field especially, if you cannot compare the data from comparable studies between centers, that there's no credibility for EV research or obtaining new grants. I mean, the comparability of data is the key to clinical implementation. And I can give you the example for flow cytometry. In 2018, we published in the lab comparison study showing that there was no flow cytometer able to measure any particles below 300 nanometers or so, or only one instrument out of 20 or 30. Since then, because we have been working on calibration and standardization, we now give summer courses on how to calibrate an instrument. Now, the people from the companies are coming to our summer course to learn how to calibrate the instruments, but they also have invested heavily the last couple of years in making more sensitive instruments. And now in the latest interlab comparison study, only 4 or 5 years later, all instruments, part 1 or 2, could measure 100 nanometers extra. So what you see is that if you create together a sort of platform that really generates comparable data and has credibility, then also the industry becomes interested in putting money in it. It doesn't make sense. If everybody keeps doing single-center studies with incomparable data and not transparent reporting, then why should you put money in it as a government or as an industry? Because it's not attractive.
Zoltán Gilis from Semmelweis University, Budapest. So I was actually thinking about what you are saying from the regulatory aspect. If I gather correctly, the trend analytical procedures really drives the differences between patient cohorts. So don't you think that just expressing way more concrete recommendations, guidelines, would help not only the science community but the regulators to ensure the quality of the multicentric, especially multicentric studies?
Yeah, I agree with you, but again, the point is every hospital, every biobank has different equipment and different operators. And what we see is that even if you give a very detailed protocol to different institutes and experienced labs, we still have plasma that is not comparable regarding the composition. So, and I don't know how to solve that problem, but I think it's difficult because probably in one lab it will be 20 degrees, in the other 23 or whatever. So there are so many variables. So I think in the end, giving recommendations there that people do measure, for example, platelet concentration simply as a measure to check the efficacy of the centrifugation procedure, I think that would be easier. I can tell you, if you go to EV-Track, I think it's a nice example. I think there are at least 200-300 different protocols people are using to prepare, to collect blood and to prepare plasma for serum. All different protocols. I'm not sure whether we can ever standardize this, but what we have seen is that even if the labs have the same protocol, there's still inconsistency in the samples. Keep in mind that from our calculations, estimations, the volume of vesicles in normal human plasma is about 0.00005%. So it's a very small needle in a huge haystack. And everything you can do to at least get some insight in the consistency of the samples that we're using as input material for omics or for plotting studies or whatever, I think that would be very beneficial. Better comparability, better reporting. as a first start. I think that would be very helpful.
I have two questions, please. The first one is about hemolysis.
First about?
Hemolysis.
Yeah.
So when you measure concentrations or even cargo of EVs from blood that's hemolyzed, I mean, do you see huge differences, or should we completely not use hemolyzed plasma samples, or use it with caution, or what would you recommend?
Well, about hemolysis again, I am unable to give any recommendation about whether you should not use or not use such samples when they have hemolysis. But at least report that if you do an omics or whatever analysis you do, report that in 5% of the samples there was significant hemolysis, or that you measured it or whatever. Because people do, at the moment, people do not report it. And yeah, so that does not help.
Apologies.
So I would use samples but report it.
Yeah. Because I had once the opportunity to have like my own blood really, one hemolyzed and one non-hemolyzed plasma, and I saw huge differences in EV concentrations between both. But I wasn't able to get this opportunity again, so I'm not sure if that's something that's consistent or not. But it might be that we should, I don't know, what should we do?
Well, I mean, it's simple. If you have hemolysis, you also have blood cell fragments or vesicles. ADP is released. That's a massive platelet trigger, so you get platelet activation, especially in citrate blood. Of course, you will have differences. But again, report the differences or do it 10 times and publish it. I mean, yeah.
I'll try. Hemolyze my own blood.
Yeah.
And Rick, I have another question about biobanks. Because there aren't biobanks that would store platelet-poor or platelet-free plasma, so I just wanted to know if we compare a blood sample or analyze a blood sample or EVs from a plasma that's stored as plasma and another from the same person stored as platelet-poor plasma. So both stored in -80 and then we analyze both. Do you think there would be differences? Because this might have some residual platelets, but that's got the full population really.
Yeah.
So I'm not
Well,
sure.
Very recently I published a paper with Pia Sanyander. It's called The Beginner's Guide to Blood EVs or something like that. It's published in JEF. And this is one of the questions we address there, I think. I'm coming from the field of hematology, and we publish now in this paper that terms like platelet-poor plasma or platelet-depleted plasma that everybody's using, including ourselves, by the way, they're wrong. They're misleading, because even what people call platelet-poor or platelet-depleted plasma is still contain platelets. It's impossible to get rid of your platelets. I can also tell you why. It's confidential because we work on the paper, but our impression is that if you centrifuge blood once, you have plasma, and the plasma contains platelets. You centrifuge a second or third time, and that concentration of platelets hardly drops. And we think, because maybe these are not platelets that are very active anymore, but it's like a sort of platelet ghosts. They may have a lower density, and by centrifugation, you're sort of selectively enriching these lower density platelets, and that's also the reason that whatever you try, they keep floating, and you cannot get rid of them. Not sure whether it's true or not. We're working on it, but yeah, just be aware of that. And that's why we also published another paper where we introduced filters that can be used to remove the final platelets. If you use these filters, the platelet concentration by flow cytometry is below the detection limit, so it's really, really low. And we think it's actually quite simple to do. If you would do that and you would freeze your plasma samples without the platelets and no signs of hemolysis, you already would have a starting material that is much better than what people use nowadays. I had a discussion before we started the meeting about the abundance of platelet vesicles in normal human plasma. We also published it for years. It's an artifact. It's just the fact that the platelets are just fragmenting in the freeze thaw process, and we measuring platelet fragments rather than platelet vesicles. David?
Thanks, Rick. we've done this recently where, as much as reporting standards and inclusion criteria are really important, we've also looked at exclusion criteria, right? So things like hemolysis is one, but maybe fat content is another, platelet sort of activation is another. And our primary readout is sort of proteome depth as well as coverage, right? So as soon as we see any confounding variable from a sample, we either have a significant impact in the depth of our proteome, so how many proteins we're seeing, as well as the coverage, this dynamic range, okay? So we've come up with a very, very simple strategy. It's applied to sort of mouse, human sort of samples, but basically it's to do with density. And yes, there are very, very good size exclusion protocols as well, but you need much greater volume, all right? So we've kind of standardized as best as we can. But the key thing is basically how consistent is a healthy cohort sample, all right? So making sure that that primary variation is really, really tight, because unless you control that, you can't really take the next steps to look at whether it's a cohort variance / a disease variance.
Yeah.
So maybe just, if you can just comment on maybe exclusion criteria.
Yeah, well, that's a great point. So, of course, it depends on the downstream analysis. I mean, plasma is still they're still fairly complex fluids. And for example, what we saw with the microRNA is that even the concentration of platelets 100-fold below the concentration that can be detected by a routine hematology analyzer disturbs the microRNA measurements of vesicles. Yeah, perhaps then you can say that if the platelet concentration is below a certain or above a certain lower limit, above a certain threshold, that you should not use these samples for microRNA analysis. I think one of the things that surprised me most when we wrote this My Blood EV paper is that apart from the fact that everybody is using different protocols, people really pay no attention at all to the quality. But if you, for example, would measure and report hemolysis and the platelet concentration, or lipoproteins, you also would be able to much better compare the samples between the biobanks. Now, every biobank has their sort of standalone system. It's like a single center study. You cannot exchange the samples between biobanks because the quality and the consistency of these samples is completely different. But by knowing, for example, the platelet concentration, if that's relevant for your downstream application, whatever it is, yeah, then you can say, I will combine samples from different biobanks. For example, if the platelet concentration is less than certain concentration. So it also gives a huge opportunity to better use and combine biobank samples, include or exclude certain samples. And of course, it's all dependent on what you're going to do downstream. I mean, it may disturb the microRNA measurements, but be completely irrelevant for a functional clotting assay. Does that answer your question a bit? Okay. More questions, ideas?
Thank you, Rick. It's just sometimes one is in a situation where there are lots of disease samples in a biobank stored as plasma, and then mostly their healthy controls would probably be people from the lab, and it's going to be probably stored in as platelet poor plasma. Do you think it's comparable? I mean, can we really combine samples stored as platelet poor or platelet free and plasma?
What do you think yourself?
No. No. But I don't know. I haven't tried it, so I'm thinking from your experience. Have you seen differences? But I don't.
Well, usually the difference in the fact how the samples were collected and handled, the differences in composition have a bigger effect in the downstream analysis than the fact that somebody is a patient or a healthy individual, unless we're talking perhaps about the present human-derived vesicles. But in many studies in cardiovascular disease, for example, people looking at platelet vesicle concentrations. Well, I'm pretty sure we also did it ourselves. No, that's the case.
Thank you.
More questions, ideas? Do you agree with this approach? I'm curious what you think of it. Who can you raise your hand if you think this is a sensible approach, or do you think this is just plain right? Be honest. I mean, I don't care. See one? It's not a good approach.
Right. Urine, you cannot use platelets.
Can you use the microphone, please?
Sorry, I was just going to say something before you started. I'm Clotilde Terry, so of course I'm trying, we have lots of biomarker studies that people want to do in our institute or with partners because I have created the EV platform at Curie. The problem I have is that we cannot force people to measure the platelets, to measure the lipoproteins. They don't know how to do it. They don't want to put the effort into that. So my feeling was that it would be more easy for us if we have, the EV platform to measure remnants of platelets, markers, or lipoproteins in the EV preparations.
Yes.
So it's a bit like the comment about the proteomics where we could see the biomarker, the markers corresponding to platelet. So to me it would sound more easy to tell people, look, in your EV preparation, measure this and this and that and try to quantify the remnants of platelets and lipoproteins and hemolysis.
Yeah. Well, actually, if you would have biobank samples and you thaw the biobank samples, supposing you're the first to thaw them, that it's not these samples that have been already frozen 10 times, most of the platelets do not fragment. So let's say 10 or 20% of the platelets will fragment. So it means that if you thaw the samples that you get from a biobank, it's still very easy with the flow cytometer. You don't even have to label the platelets. You can very easily quantify the platelet contamination. Hemolysis is visible. You can just see by eye whether and there's even a sort of simple, you have these strips with all these color codes. You can sort of semi-quantify the extent of hemolysis. I think already, and if the plasma is very fat, you will also see it immediately. So I think you can already do a lot also yourself. Because it's a starting material for your omics, it's very easy to try to measure the platelets.
Can you hear me? No. Okay, when we're doing proteomic analysis, I think before even we look at a patient to a healthy cohort, we need to understand the dynamics of pre-analytical variables before we go downstream. So our initiative was to look at how does postprandial fasting, which time of day you take your sample, and especially for the urine samples, which is it a random urine, is it a 24-hour, because this impacts the data that you're going to compare. Sure. And if you're going to compare a sample, for example, in most of the cases, hemolysis versus non-hemolysis, it's just not comparable. Because within our healthy cohort, we have such a great biological variation, as you know, and to add on to that, the technical variation. So if I'm a proteomics person, so I want to look at the pure biochemistry changes before I actually look at my patient difference. That's one of the things. And the other thing is that when I look at proteomics, I use EV as so-called cleanup process because it removes the albumin that needs to be removed. And you work with saliva and you need to remove the amylase. So urine, you need to remove protein that is there. So all these kind of things needs to be addressed within this EV. It would be nice to bring some of these approaches in. So, because we have published papers before looking at how the platelet count changes with every centrifugation. There's many papers on that. But if you want to use, for example, a urine sample, and you're doing, you can normalize against the urea creatinine. But if you're not in a hospital situation, that becomes a difficult task, right? So you need to be linked.
Yeah, so about this biorhythm, of course, you have a very important point. But I think as long as we have the analytics, like simple preparing the starting material are not in place, we still have a problem. So I would think report as much as you can. That's often the recommendation. And perhaps with hindsight we can look back and see what the effect is when you collect samples in the morning or evening or night. So it's an important point, but I have no clue what to do with that right now. The other question was about what did you say? Oh, urine and other body fluids. Well, I'm not sure. I think the most important thing is that is becoming a sort of awareness in the EV field that measuring EVs is difficult, isolating EVs is difficult in detection. Right now, there's not a single method that can isolate or detect all vesicles or exclusively vesicles. It's very, very challenging. It's really very challenging because the vesicles are present in a soup of other stuff. And at least if you're aware that that's the case, and if you try to have the samples, your starting material, as pure as possible, or at least know the quality and report the quality of samples, I think that would be very beneficial to get to the next step, next phase. And whether it's urine or plasma or CSF, I think all these fluids is the same. I've reviewed papers for journals where people using lymph and they saw a massive concentration of platelets and red blood cell vesicles, and they said they had no blood contamination. But after 3 reviews, finally they said 3 rounds of review, they admitted that they had blood contamination. They reported it, and I thought now the paper can be accepted. Just be transparent about it. Just say that the sample is good or not. I think it's nearly time. If there's one final question, if not, yeah, just one short final question out.
Yeah, sure. Thank you so much. I appreciate it. So, what is a kind of ideal situations in terms of the biomarker using the blood in the futures? So that's the things, situation we wanna pursue in the future. That's the thing.
Well, it's a nice question. So, if you've been in the EV field for a while, people have been studying about the huge biomarker potential of vesicles. And if you're very honest, I think we're under 4 or 5 small, pretty small, not really clinically well-established applications of extracellular vesicles. I think the incomparability of data is the major cause of that. So I still think there's a huge potential, but as long as we all keep doing our own stuff in our own lab and generate data that cannot be compared to one of the neighbors, we do have a problem, and there's no clinical use or utility. So I think if you really want to explore the biomarker potential, really clinical relevant biomarker potential, then you should pay much more attention to all these steps. I think that's very simple. Yeah? Okay. Well, thank you for your attention, and let's try to get a cup of coffee.